Well, hello, folks. So the exam is over. Second exam, you start in the third exam's material. Kind of started that in lab this week. I'm not going to be around Monday and be out of town, and so I'm going to go ahead and record this lecture as kind of not a supplement, but the actual lecture that I would normally do, beginning the respiratory system. So you should watch this and take notes and take it seriously and consider this the same thing as a lecture. So the material in this is going to be fair game for the exam. My hope is to get through this today. It would be equivalent to what I would cover on, say, Monday if I were there for a regular lecture meeting. So Wednesday, I'm going to pick up where I left off in this lecture. All right, so I'm not going to go back through all this stuff. So you should know that. I've been fielding questions about lab and lecture grades and so on. Recall that the lab grade is 30% of your grade overall. If you didn't do so hot on this last practical, you got two more to make it up. I will likely do some grade adjustment. So, I mean, take heart in that. Now, if you're really just dropping the ball altogether in lab and putting all of your eggs into the lecture basket and doing better there, that is a bit of a problem. And you need to do what you can to rectify that. And that would entail just simply a whole lot more study and review and so on. So some of you just nailed the practical to the wall, and I was really proud of that. And others of you kind of just let it go. I'm not sure what was going on there, but anyway, we got a significant amount left in the course, and I'm confident many of you are going to do what it takes to perform well. Anyway, let's go ahead on into this lecture that I want to do on the respiratory system. All right, let me see. I'll share this. All right, I got to do some little display setting things here to make sure that this is as it should be. Now, of course, the Emmerman textbook has the respiratory system in a different chapter, so pay no attention to that. We're on the respiratory system, okay? And I'm using these PowerPoints from another book because I like them and I'm familiar with them. So anyway, we're talking about the respiratory system, and what we'll find is perhaps for you, it covers a surprising amount of different kinds of activities in the body that help with maintaining homeostasis. I think everybody knows that when you breathe air in, that you can extract essentially the oxygen from it, and that passes into your blood. And it's, when that happens, we say there's an exchange of the oxygen for carbon dioxide. So carbon dioxide is passed out of the body. And so a lot of people will list that as the premier function of the respiratory system, is this exchanging of gases that happens. But along with that are a variety of other things that we'll touch on as we go. Not the least of which would be pH balance, maintenance of pH in the body fluids, and the breathing movements we've touched on help with movement of lymph and venous blood returning to the heart. And there are even other functions still that we can attribute to the respiratory system. So it's incredibly important. Now, when respiration happens, when we use this word respiration, you should know that the meaning of that varies depending on the context in which we're using it, okay? A lot of times we talk about cellular respiration, which only in a very indirect way is related to the movement of air in and out of the body. Cellular respiration has to do with the production of ATP, this high energy molecule, using oxygen, using food molecules like glucose, and processes like glycolysis and the Krebs cycle and electron transport chain. Those are all molecular kinds of processes that are happening and are not, strictly speaking, the same as respiration in the other sense that we use that word. We should rather talk about breathing, not as respiration, but as ventilation. All right. When we breathe, we're ventilating. We're bringing in air. And then, of course, exhaling as well. External respiration has to do with the movement of the air into the airways. As we go down into the lungs, we go into bronchi, and those branch repeatedly to become smaller and smaller and more numerous. Finally, we go into bronchioles, and at the very ends of those bronchioles, we have alveoli. All right, and so there we'll have the gas exchange occurring. The oxygen goes into the blood, CO2 comes out, and now the oxygen in the blood is going to be transported around the body. Since it's bound to hemoglobin, it's carried efficiently that way. It is then unloaded at cells, and so that's internal respiration. Cells take it in and exchange it for carbon dioxide. So we've got a little bit of gas exchange at the cellular level in all of the different parts of your body. Then, of course, the oxygen will be passed on into structures that we utilize it in cellular respiration. So just know that flippant use of the word respiration is probably not a good tactic, that you should replace the term ventilation for the process of breathing, of taking air in and out of the body. And that, respiration has all of these other things that it can mean. All right, and so we're breathing so that we can accomplish gas exchange, bringing in oxygen, getting rid of CO2. And the ultimate goal of that is to make ATP, as we pointed out. The CO2, if it builds up appreciably, can be problematic. We need a little bit of it to help with maintenance of pH. The rest is going to have to be eliminated and blown off into the surrounding environment. Okay, so that's primarily why we breathe, is to accomplish those things. So we want to go through the parts of the respiratory system, and it's quite extensive. A lot of times, the people who teach courses like this will kind of do a cursory coverage of the things up here, the nose and the nasal cavity and these sinuses and so on and so forth. And when they get down into the lungs, that's kind of where they emphasize things. So I'm going to try not to do too much of that. I want to make sure that we don't completely dismiss all the stuff up here. All of the stuff up here is part of what's called sometimes the upper respiratory tract, including the nose, all right, which is more than just a sort of an ornament on your face. It's got these openings, these apertures called nares, and they lead on into the nasal cavity. Then you got sinuses. There's several of these. They're sometimes referred to as paranasal sinuses. They are simply places within certain bones that are lined with mucosa, there can be some secretion of that mucosa to, lead to some fluid accumulation there. Sometimes it becomes excessive and you have a lot of pressure. But these sinuses help to reduce the weight of those bones, and also because they're going to kind of vibrate a little bit as we produce sound, they're going to help with amplification of sound. So when you're speaking or singing or whatever you're doing, that's in part what's going to help you to project a little bit is the fact that you've got those sinuses in there. So there are sinuses. The one here, right between your eyes, essentially just a little bit above that, is the frontal sinus. And then you've got in the sphenoidal region, the sphenoid bone sitting back in here behind where your eyes are, back near where the pituitary gland is located, you got the sella turcica. And just above that in the sphenoid bone, you've got a sphenoidal sinus, and there are maxillary sinuses in here, variety of sinuses in different places. So the pharynx, when we use that term, remember that's referring to the whole sort of posterior region here, back where, you'd have inflammation if you had, say, a strep throat or something like that. When your tonsils become inflamed, when the adenoids become inflamed and so on, that's in the pharyngeal region. And we'll talk about how it's divisible into different sub-regions based on, landmark structures around there, and also functionally. I mean, there's a little difference among these different parts of the pharynx in terms of how they function. And the epithelium varies from one place to the next. So that's the upper respiratory tract up down to about here. And then below that, we're into the lower respiratory tract, which includes the larynx, this set of cartilages, including the thyroid and the cricoid and corniculate and other cartilages. The epiglottis would be considered part of that whole assembly. That structure is there to keep the airway open, to keep it from collapsing, and to help with the passage of air and food past one another to ensure that that's done efficiently. Anyway, that's the beginning essentially of the lower respiratory tract. And then we've got the trachea, which is unmistakable because it's this long single tube that has these C-shaped, as we learned in lab, C-shaped cartilages. Hyaline cartilage is what that's made out of. And then we get down into here, we got these branches that lead off into the two lungs, right? So those are bronchi, and those bronchi, as they penetrate the lungs, will branch and then branch again and a branch again. And in fact, if we count the number of branches, it winds up being on the order of about 23 branches, or 23 generations of branches, which, is a tremendous amount. And the tiniest little tubes that arise all through the lung tissue out here, you'd need a microscope to be able to see, and they're referred to as terminal and respiratory bronchioles. Those lead into the air sacs, the alveoli that are going to be used for gas exchange. So the lower respiratory tract includes all of that. By the way, because of this branching, the set of bronchi and bronchioles that you have in these different lobes of the lungs are referred to as a bronchial tree. So here's our upper respiratory tract, at least down into here. And so this is a kind of a lab thing, so I'm not going to dig in and really go into great depth on this, but this nasal cavity has different kinds of epithelia in it. Here in the beginning, right around the nares, it's stratified squamous epithelium. But as we enter back into here, it's a very thin, sort of moist, serous kind of membrane. called a mucosa, and it's mostly respiratory mucosa, all right? Mostly respiratory mucosa. Although right about I'd say about in here is mucosa that's adapted for smelling. You have olfactory neurons that project down to that area. This is where the ethmoid bone is, and there's a little plate in there through which olfactory nerve endings project. And so the epithelium right in there is considered an olfactory epithelium, but the rest of it here and then beyond that is respiratory epithelium. In fact, a respiratory mucosa. In fact, that is what we have in here, down in here. No, not right in here. This is the oropharynx. That would again be stratified squamous epithelium, a little different than that which occurs right here about the nares, in that this is going to be non-keratinized, which means it's not toughened or hardened, waterproofed with lots of keratin. We don't expect that to happen there. Right here, proximal to the nares, for a little ways in to the nose, we do expect a little bit of keratinization and some hair and so forth to be present there. These vibrissae, little stiff hairs that rim the nostrils, help with filtering air. So yeah, respiratory mucosa in here, and then in here, and then you've got oropharynx, a little bit of olfactory mucosa right in here. Now these scrolls of bone that are covered with mucosa right here, those are called conchae, C-O-N-C-H-A-E, because of the scroll work of bone within them that I talked about in lab. And they're there to make the air turbulent. As the air passes into the nasal cavity, it sort of mixes and creates these little whirlpools and eddies and so forth. And the air just kind of hangs here for a little while, giving it adequate time to be moistened and warmed, so that when it goes back into the lower respiratory tract, all the way down in here and goes into the lungs, it'll help the lungs expand easier. And also, due to the fact that it's moistened, won't crack any sort of membranes down in there and lead to the possible entry of pathogenic microbes and that sort of thing. You do have some tonsils that are positioned in here. That's lymphoid tissue, but just as landmarks, you should know the lingual, the palatine, and the pharyngeal tonsils up here, the adenoids, are located right there. Okay. And I did mention earlier the pharynx is divisible into 3 parts. So this part here, you can see the soft palate in the roof of the mouth. This palate separates the airway from the buccal cavity where food is chewed. They're completely separate, so we're not going to have prevention of breathing while we're eating, essentially. The region just beyond this soft portion of the palate, right in here, you can see a landmark would be this opening of the auditory tube that connects the middle ear region with the pharynx, so that materials can come in here and help to kind of take pressure off of that space. In that region right there, we've got the nasopharynx, the so-called nasopharynx, which is equipped with a respiratory mucosa, having cells that are ciliated that can be, in some cells, goblet cells for production of mucus. And that's going to help to clear out any debris, any pathogens. The cilia beat, the mucus captures this stuff, the cilia beat, and drives that material down in here where the oropharynx is. And you'll then be stimulated to swallow that. Just the presence of material there triggers that swallow sort of reflex. The Oropharynx is a different epithelium. It's got stratified squamous epithelium, which is going to be protective against tearing and mechanical stress. The laryngopharynx, you can see the epiglottis here in the larynx assembly. The epiglottis, the region adjacent to its tip, and then on down to about here, where we have the rim of the trachea back here, the trim of the rim of the larynx. That region right in there corresponds to the laryngopharynx. And we have, once again, on through here, the reappearance of respiratory mucosa, these ciliated columnar cells. It's actually a transition, or not transitional, pseudostratified type of epithelium where the nuclei of the different cells are at different levels, giving it a layered look. It's not really layered. But yeah, that's what we have here. And the cilia in this region, when they beat, are going to beat upward and drive the mucus in that direction. So that's incredibly important to kind of keep your lungs clear of potential pathogenic substances. All right, so yeah, this is just a little bit more on the nasal cavity. Gives the landmarks around it. And you got your hard palate, and below it, the cribriform plate is the fancy name for that region right there, where we've got the olfactory nerves coming through to synapse with cells in this olfactory mucosa right here. That's the cribriform plate. That marks part of the upper sort of ceiling above the nasal cavity. And we got a septum that goes through and divides the cavity into left and right sides. If you've ever looked, into the nasal cavity area of a skull, you'll likely have seen that. I'm thinking I might have a skull here in my office. I don't. I sometimes have other mammal skulls that, principally the same as ours in a lot of ways. But there's a septum that is partly composed of cartilage and then partly bony the further back we go. Yeah, and then here's, a little bit about the mucous membrane. Notice how mucus is spelled, by the way. When we describe a membrane as a mucous membrane, we use this spelling. But when we're talking about the stuff, the actual mucus, which is protein mixed with water, that forms this sort of goo that can capture, particles that are floating in that are being breathed in. When we use that word, it's MUCUS. That's the actual stuff that is mucus. All right, and this mucous membrane is mostly pseudostratified ciliated, except for the olfactory membrane, which is not like that. So air is hopefully warmed, moistened, and filtered efficiently as it comes through that passage. There is the pseudostratified ciliated epithelium. The little hairs here are cilia. These cells here that are kind of scattered in amongst those cells are goblet cells. All right, you can see it looks like a cup. The membranes come up either side, and you can see this little vesicles containing the protein mucin, M-U-C-I-N. That protein is being released to the surroundings, and at the top here it just sort of spills out, and as it mixes with water that's condensing there, it'll form a secretion that sort of coats this whole area. So you got all of this mucus accumulating, and here then are pieces of debris being caught in it, germs and so forth. This down here is simply fibrous connective tissue. It forms what's called a basement membrane, right? And in the trachea, actually, when we get down below this, we'll see additional glands there. This is down in this region would be a submucosa. It doesn't show it in this picture. Over here, it kind of does. Down in this area would be submucosa. Yeah, that's what that is. And there's some glands that occur down in there that are subsidiary to, or accessories to the goblet cells here that are producing lots of mucus. So mucus is really valuable, really important stuff, because it helps to condition and moisten and capture potentially infectious particles that are making their way in. So yeah, here it's talking about the sinuses. I mentioned the frontal, maxillary, and the sphenoidal. I forgot there's also an ethmoid sinus. So again, these are places that are just sort of space that helps to reduce the heaviness of the bone in the skull, so that your head's not flopping over to the side all the time, and also they help to make the production of sound more efficient. They help with phonation and provide some resonance to kind of amplify the sound. All right, yeah, so this shows a skull from the anterior perspective, and you can see these spaces back in here in the skull would be some sinuses that are present there. Yeah, it's shown up here in this picture. You can see the frontal. There's the sphenoidal. All right, these are maxillary sinuses that when they get full of fluid, say during allergy season or when there's an infection there, which can happen because they do have connection with other areas that ultimately can connect with the outside. When they get infected, especially these right here, these maxillary sinuses, it'll put pressure on your teeth. And some people remark that, they're so sick, if they get a head cold or something like that, they're so sick that their teeth hurt. That's not an exaggeration. That's exactly why that would be. And actually here, this is partly nasal cavity up in here, but there's partly also maxillary sinuses present there. All right, there's the pharynx again. We don't need to belabor that. I've already talked about it. Here's the larynx, both anteriorly and posteriorly. So you've got associated with this a hyoid bone, which is actually part of the skeleton, but it helps with swallowing and other kinds of movements. You can see it's connected to the larynx beneath it by way of this, what's called sometimes the thyrohyoid ligament, this sort of membrane here. Here's your thyroid cartilage with this laryngeal prominence. This is a point that pokes out. That's the Adam's apple. You've got a cricothyroid ligament here that binds these two together, the thyroid and the cricoid. And you can see on the backside, the cricoid cartilage has some extensions that come up. These are actually called corniculate cartilages. They're not named, or these are corniculates, and there's the arytenoids is what they are. That's kind of very specialized information, and outside of knowing what those are when you hear them, I'm not going to have you know much else about them. This is the epiglottis, which you can see a little bit of poking up above the hyoid bone here. You can see from the posterior view that it's sort of shaped like a spoon. And when you breathe, or when you swallow rather, the whole larynx sort of shifts upward, and this epiglottis is sort of flexibly attached down here. It can come down and, like a lid on a pot, just sort of cover that. And the good thing about that, obviously, is that material that's in your mouth that you're, say, drinking or eating, when you swallow, won't make their way down into the airway. So that's the structure that helps with that. Actually, you can kind of see a little groove here between these two arytenoid cartilages. When you swallow, the epiglottis slides down, and the base of it actually fits in that little notch, very, just like that, and seals that off much better than you would expect otherwise. All right, and there's the larynx from other perspectives. And so, yeah, here's the epiglottis, which you can see is covered with respiratory membrane. We usually show it as a cartilage stripped of that, but it's covered with that membrane. And then below that, right in here, is a little area through which air can pass when it's wide open, when it's patent. When you're, taking in air and holding it to brace your core for like lifting or something like that, or you're performing the Valsalva maneuver, as we've talked about previously. When you do that, you close this off, and the muscles in here will kind of pinch that closed, and the air is held in tightly. But when you're breathing, this little entryway here can open. It looks kind of like a slit. It sits below the epiglottis, and so we call that region the glottis. And right above it, set in its walls, are a couple of little shelves of some mucosa covered tissue there called false vocal cords. And then if you go down a little bit below that are some other structures that poke out that can vibrate in response to air passing over them. And that's how sound is produced with these true vocal cords. They're positioned down there. All right. So the false vocal cords, their job is not unimportant. They actually help when you're swallowing to kind of come together and seal that opening off much better. So that, along with the epiglottis, is going to help prevent aspiration of things that could possibly block the airway. All right. And so, yeah, this shows the glottis when it's closed, and it names the cartilages around there. There's your epiglottis right here. All right. And here is a true vocal cord. All right. And set above it is this more mucosa covered looking false vocal cord. It's above the true vocal cord. Okay. So know that those are present. Now, if we follow down into the tube beyond, it's reinforced with cartilage as well to maintain patency. It's a term that I used in lab the other day, patency. You've got these cartilaginous sort of what appear to be rings here. They're actually C's. They're shaped like a C. They're open on the back. A muscle covers that opening. The trachealis muscle is back there. So there's that trachea, and it gives dimensions of it here. It's about 2.5 centimeters in diameter. A centimeter is about the width of an aspirin tablet. So that's usually what I've pictured in my mind's eye to kind of make sense out of that, because we don't use, a lot of us don't use metric measurements in our daily lives. In scientific investigations we do, because in actuality they're much easier to convert from one unit to the other. Anyway, 2.5 centimeters, which is about an inch. I think an inch is 2.54 centimeters, so that's right in that ballpark. So it's an inch across from side to side. And then in terms of length, it's about 12.5 centimeters, which is 2.5 goes into that for, that's about 5 inches. Okay. So it's not terribly long. All right. The larynx is going to take up some of that distance here. And then you got your trachea, and then it branches. Now this branch point right here, this place where, you know, you've got this sort of crossroads that you come to, is the carina. And it's significant, not simply as, you know, a landmark, but the lining of it, the respiratory epithelium in it, is very sensitive. There's some nerve endings that project into that region that when they are stimulated by tactile kinds of pressures, that is things touching, as would happen, say, if you aspirated something, you know, like a a seed or a bit of some kind of food or even some fluid, if it settles in there, that's going to trigger really violent coughing to help kind of clear that, to push it back up. And, you know, that which you don't get coughed up, you got small residual things left behind, the respiratory epithelium will catch that and beat, you know, the mucus on it will be what will catch it, and the cilia will beat to drive it up out of there, where then you're going to swallow it. Okay. And that would take it down into a inhospitable, very caustic environment inside of your stomach where there's a lot of very powerful acid. So whatever it is that you swallow, they're going to get destroyed there. Which is, you know, peace of mind is what that gives you. It's like your stomach's going to make sure that whatever that bug is, whatever the virus or bacterium it is, it'll pretty much just degrade it and tear it down to nothing. Now you can see where the trachea branches here and goes off into either lung. So we've got a right bronchus. It's described as being primary because it's the first one. And there's a left primary bronchus. And between the two, this right one actually descends a little bit more steeply vertically than the one here on the left, which isn't quite as steep. And so when you do aspirate something, this is the side usually where it's going to go. And you can see beyond this first little primary bronchus, it branches again into two. And then this branch here branches into two. This branch here will branch into two. And then, you know, you can follow those branches out and see that each of those in turn will branch again and again and again and again. So all of these are called bronchi as well. A bronchus is a branch off the trachea or some branch thereafter that is held patent by the presence of cartilaginous rings, or as you get further in here, just plates, little pieces of cartilage sort of wedged together like pieces of a puzzle to create a tube. And so those would be called bronchi as well. When you run into areas where it branches so extensively that we can no longer fit cartilages in there, and frankly, we don't need them because they're so thin that they're not going to collapse under their own weight. We're talking about what are called bronchioles. So bronchioles are very tiny and very numerous. There's lots and lots of them. We couldn't even, you know, count them typically. That's how abundant they are. And there's tens of thousands of them. So yeah, this passage in through here, all of these tubes, the trachea, the two primary bronchi, the secondary, the tertiary, you know, and bronchi beyond that are going to retain this ciliated sort of appearance. You're going to have a pseudostratified ciliated epithelium. Eventually, you're not going to have enough diameter in the tubes beyond that to maintain the pseudostratified appearance. Instead, the cells will be cuboidal. They'll be little short, squatty cubes. And ultimately, you know, that's going to give way to a simple squamous epithelium in the very deepest depths of all of this. All right. Note too that the lungs have multiple segments in them, or lobes as they're called. So the right lung, which is this one over here, has 3 different lobes in it: a superior, a middle, then an inferior lobe would be down here. And you got these bronchi that serve those lobes. There's actually a superior lobe bronchus. It goes into that lobe. This is the middle lobe bronchus, and then there's an inferior lobe bronchus that goes down here. On the left, this is the left over here, there's actually just 2 lobes of the lung, set off by these sort of cracks in the surface of the lung, what's called a fissure. It looks like a little crack in there. It's a little groove. You've got the superior and the inferior lobes on this side. Actually, there's a little bit of the the fissure that you can see in the left lung. All right. This shows a tracheal cartilage, one of those little rings. This is the respiratory epithelium in here, which has goblet cells in association with the pseudostratified ciliated epithelium. But down beneath that, in this what's called submucosal layer, you can see some glands in here, and those contribute even more mucus. And so there are some tracheal glands. If that's not just an artifact of how they drew it, those are real things in there. And so they add more to the mucus, which is helpful. All right, this shows here a little incision. This might be something that would be done if, say, a person had massive swelling up in here that prevented opening of the glottis to allow air to enter, and that sometimes happens. You've got injury in the upper respiratory tract that prevents air from coming through and entering the glottis. So here there's a little incision that is made right through the skin and the underlying tissue. There's not a tremendous number of blood vessels there, so there'll be some minimal bleeding, but for the most part, if you can get through there with something, you'll potentially save somebody's life, because that provides an airway now that's patent. You got to keep it clear, but that's why that's done, and that's called a tracheotomy. Let me try to use. I've got a pen that comes with this. There's this device I have called a Wacom, and hook it up to the computer, and it's supposed to make it so that I can use devices right on these PowerPoints. And there, it's not working right. Let me go back to it. Let me see. It's called tracheotomy. Oh, gosh darn. There it is. I don't know why it does that. And then these little lines show up on there, which I apologize for. I can actually blank this screen out and pull up another little whiteboard, but that's extra keystrokes and kind of a pain. If I have to do it, I will, but right now I don't need to. So anyway, that's called tracheotomy. And there have been people who know how to do that, who've had nothing more than like a sharp piece of glass or a pocket knife or something that they have cut through there in order to open up an airway to allow somebody to breathe. That maybe they're dealing with anaphylactic shock or something such as that, and their airway's closed off, and that will allow them to continue to oxygenate their blood. All right, and there's the bronchial tree. These different lobes here, the superior, there's the middle lobe, the inferior lobe on the right. On the left, we have just two lobes, superior and inferior. And here are the fissures. That's a horizontal fissure there. These two down low are oblique fissures because they're running diagonally, or obliquely is the term that we would use. And so these bronchi divide and divide and divide, and then eventually form bronchioles. And the actual final little segment, which is so tiny, it's microscopic, it's called a terminal bronchiole. And if you follow a terminal bronchiole far enough, you'll start to see these little air sacs arising in it, just single individual air sacs poking out. And that would mark this as a respiratory bronchiole, okay? And that's a site where you can have, for the first time, gas exchange. All of the parts of the respiratory tree above this bronchial tree above this, are just for conducting air. So we call that the conducting division. But way at the very ends, you have tremendous numbers of respiratory bronchioles. And then you see individual sort of single alveoli coming off. And then finally, at the very ends of these, you'll have these whole alveolar sacs, right? And this is a duct leading into it. So that's called an alveolar duct. And all of these membranes, including these singles out here, but these big sacs are more important. These are composed of simple squamous epithelium, very thin. And there's some fluid that's being produced by certain cells that we'll look at here in a little bit that accumulates along the walls of these sacs internally. That helps the sacs to be permeable to gases and also to keep them from collapsing. Yeah, so that if you count all of these alveoli, you got like 1 million per lung, all right? And you've got capillaries that surround these. And so the capillary wall and the alveolar wall constitute a kind of epithelium across which gas exchange can happen. We'll look at that. But yeah, there's the whole bronchial tree. And what they've done is they've color-coded different so-called bronchopulmonary segments. So, I mean, not only does the lung have these different lobes, but within each lobe, you have compartmentalized areas called bronchopulmonary segments. And the value of that is that if, say, you got an infection in this yellow region right here, well, you're going to have some tissue around it to kind of wall that off to kind of keep that segment from having an undue influence on the surrounding segments. All right. So if you have some infection or some other problem in here, it doesn't necessarily have to spread into all these other little segments. You can see just tremendous amounts of branching. This thing looks, you know, kind of fuzzy. It's got so many little tiny endpoints. And all of those are going to lead into alveolar sacs. All right. So yeah, here's all the things that you're going to encounter along the way. The right and left primary bronchi, the secondary or lobar bronchi, all right? These guys in here, 1, 2, 3 on the right, 2 on the left. And then within each lobe, you're going to have further branching to form these segmental bronchi, all right? And then once you get past those, you're into bronchioles, right? And these are tubes that are really narrow and they no longer have the cartilage in them to support them, right? And there's very large numbers of those. They divide again, until finally you're into what are called terminal bronchioles, which give way to respiratory bronchioles with individual alveoli on them. And where these end, we've got alveolar ducts leading into alveolar sacs, little collections of alveoli. Here's a more complete sort of view of a terminal bronchiole, right? And it leads into these respiratory bronchioles. I know this is respiratory because there's these scattered alveoli that are starting to show up. These could be sites for gas exchange, although much more gas exchange is going to happen where you got a bunch of alveoli clustered. This is an alveolar sac. And this one, they leave the capillaries on it to shroud it, to show how they're arranged. And you can see every single one of those is covered with a little net of capillaries. These guys would be as well, just would clutter up the diagram a whole lot. So you got afferent blood coming in, and then it goes through the capillary beds. And then on the backside, the efferent flow is going to go out by way of a vein. And obviously that's going to carry the blood out to the heart, where the heart then can put it back under pressure and blast it around the body. All right. Actually, that I had the blood flow backwards. The vein has it coming in. It makes better sense. The vein comes into the capillary beds, then it's oxygenated, then it leaves, and then that's going to go to that's going to go back to the heart. So this would be one of many millions of these that collectively would converge and finally form pulmonary veins, which are some of the only vessels that are veins that carry oxygenated blood. Simple squamous epithelium. Now you can see inside of the alveolar sac, there is this little chamber here. And then at the end, at the limit of that, where you're starting to go into the air sacs, there's these little tiny pores. And there's actually pores not only going from the air sac lumina into the alveolar sac, the alveolar lumina into the alveolar sac, but also pores that connect the alveoli together internally. All right, so what that means is if you've got one of these that's diseased and damaged and scarred, maybe not doing its job, the air that goes into it can pass into other alveoli, which can process it, which can get oxygen out of it. So even if there's some damaging, damage due to, say, emphysema or something like that, certain of these could get knocked out and damaged, but the air that's in them doesn't necessarily stay there. It's going to go into other air sacs that can process it. All right, so this kind of is a text to kind of explain what I just explained by, speaking it, telling you that. And so this shows one alveolus. Multiple ones of those are alveoli. So this is one alveolus, say in a respiratory bronchiole, all by itself. And it shows the epithelium, simple squamous epithelium. It shows capillaries around it. You got the venous, or this is actually arterial blood. But remember, in the pulmonary circuit, the arterial blood is deoxygenated. That's not typical of the rest of the body. So deoxygenated blood comes in, and as it passes through, oxygen that you've breathed in that's abundant in this alveolus is necessarily going to be able to it's going to tend to, I should say, make its way into the blood, because there's a gradient. There's low oxygen here, there's high oxygen here. That oxygen is going to make its way in the direction of lower oxygen. It's going to go downhill into that low oxygen environment. Likewise, carbon dioxide, which is abundant in this blood. Remember, this blood came from all the organ systems in the body, and so has accumulated a lot of CO2. That CO2 can diffuse in the other direction from the blood back into the air. And so as that blood goes along, you can see its color starts to change because it's taking up more and more oxygen and getting rid of more and more CO2, till finally we're into the blood at the end of all of this that's really highly oxygenated. And eventually that's going to make its way into the pulmonary veins, which are going to return it to the left atrium and then on into the left ventricle so it can be pumped out of the body. So this in here, this is the alveolus, the simple squamous cells in a single layer. There's a little bit of fibrous connective tissue and fluid here that lies outside of that. And then, of course, there's the capillary wall itself. Gases have to cross all three of those things. All right. And so there's that's going on. That poses some special challenges, but these cells are so thin that typically adequate oxygen does diffuse through them. Now, one problem that has to be overcome is the fact that when this is not inflated with lots of air, there might be a tendency for it to kind of pucker up and collapse. And that sometimes does happen but usually not. We'll talk about what it is that ensures that that doesn't occur. But yeah, so here is a blood vessel. This would be like an arterial coming in. And then you can see all the branches coming off of it. These are capillaries. And they're wrapping around lots and lots of alveoli in alveolar sacs. And you can see alveoli poking their heads out from underneath all of this. So these are air sacs. These are alveoli down in here. Air's going to be breathed into those. And as the oxygen diffuses out, it enters into these capillaries. And they even color code this to show where the venous blood is and the oxygenated blood would be. This is a section through a bronchiole. No, it's a bronchiole because there's no cartilage in it. Okay. And off of that, then you have all of these alveoli out here. So this is like an alveolar duct, or it could be a respiratory bronchiole passing into an alveolar sac. And these are the alveoli. Notice how in their walls, even there are little pores. So air in an alveolus doesn't just inflate that one alveolus, but can move through the walls into surrounding alveoli. All right. And that accomplishes this, function of passing good, high quality air into an alveolus and finding it doesn't work. It can go on to one that does. All right. That's going to help make things a little more efficient anyway. All right, so here it shows where the lungs are located, on either side of the mediastinum, within the rib cage. You can see the points of the lungs here called apices, apex. They poke up above where the clavicles are. So that little space right there actually has some lung in it. So if that bone were to break and snap, and sharp shards of it go in here, it could potentially damage the lung. All right, which is why one of the reasons a clavicle fracture is not always just a simple thing, but it can have some complications associated with it. There's the lungs from this would be a transverse section, so cutting across the thorax helps us to see how the surrounding tissues are arranged. And you can see here's a lobe of the lung, here's a lobe, and this shows a fissure penetrating in relatively deep in that lung. So this would be down low. This would be well below where the apex is. You got to be deep enough to where you can have evidence of fissures in the lung. And you can see there's a blue sort of band all the way around here. And that, it turns out, is fluid filling a space. And that's called pleural fluid. I don't think it is even named over here. But there's pleural fluid, and it's produced by the membranes that we have on either side of that little space. Little space is the pleural cavity. The fluid in it is pleural fluid. And then the green membranes outside of that are important. These are actually called pleura. Visceral pleura is right on top of the lungs. Parietal is just inside the thoracic cage. And in life, they're going to be pressed very tightly together, but that fluid will be in there. And the fluid is full of water. And water has, the ability to hydrogen bond to surfaces. And so those two pleura are going to kind of get stuck together by that water. They're literally sort of polarized and are attached to one another in much the same way as two sheets of plastic that are wet when pressed together will tend to cling together. And so the value of that is it provides a protective sort of barrier that some viruses and bacteria and things have a hard time breaching. But more importantly is the water that's hydrogen bonding to these two walls will get them to kind of cling together. The outer surface of the lung sort of clings to the inner wall of the thorax, so that when this moves, if the rib cage expands up and out with the contraction of intercostal muscles during, say, inhalation, breathing in, the lungs are going to be pulled right along with the ribs expanding outward. So that's going to help you breathe. Incredibly important. Okay. All right, so there's parts of the respiratory system, the upper portion anyway. All right, now we want to get into breathing. This is, the mechanism for it, how it happens, right? And it's a physical process. It involves some muscle contraction. What we do, by the way, is called negative pressure breathing, as opposed to, say, something like a frog would do, which is positive pressure breathing. So we use negative. Let's see if I can do this without one of those lines showing up. Negative pressure breathing. That's an A in there. Mammals can do this. Pretty much it's limited to mammals for the most part. What birds do is actually much more efficient. They're able to take air in. I guess it's a negative pressure in birds as well, but they can take air all the way through these bronchioles into lungs, into some compartments on the backside of the lungs. They can store it there temporarily, and then they breathe out, and that's when gas exchange happens. It's really a weird but very, very efficient mechanism. And birds, of course, have a much higher metabolic rate than mammals, and they engage in a very much more taxing activity. That's flight. Flight is very, very tough. It's very expensive to fly energetically and in terms of oxygen demand. So what we're talking about here pertains to mammals, especially humans, and it's called negative pressure breathing. And it relies on negative pressure or a vacuum, right? That's another term for that. And it's generated here in the chest cavity, all right? So we got two kinds of movements. We talk about inspiration, that's breathing air in, or inhalation, if you want to use that term. Expiration is the pushing of air out, the blowing of air out. And so what happens during inspiration? Well, excuse me, we generate a vacuum in our chest cavity, and these lungs will fill more or less passively as a result of that. How is that done? These syringes down here are used to sort of provide an analogy to what's happening. You've got a syringe and you start pulling on the plunger, the volume inside of here increases, right? The space gets bigger, and the pressure within that region gets lower, so that if there's air in the surroundings and it has any way to enter into that space, that's where it's going to go. This is a low pressure or negative pressure area. The air is just going to passively rush in and fill this space. Whereas if we take the plunger and push back the other way, we build pressure up in here. And that's going to cause a reversal of the flow of air, and it's going to actually make its way out of the syringe. So transferring that explanation to what happens here in the chest cavity, in a person's chest, when there's inspiration, when they breathe in, this diaphragm acts in a way analogous to the plunger on the syringe. The diaphragm, which is a skeletal muscle, by the way, you can voluntarily control it. So it qualifies as skeletal muscle, both because of that and the fact that it has skeletal muscle fibers in it. When you go to inspire, that diaphragm contracts and literally flattens out. So we can tell that this one, because it's got that dome shape to it, this person is more or less at rest. All right, they're in between breaths. Now, when that diaphragm contracts, it's going to pull all the way down here. It's going to flatten out, and that's going to give that much more volume to the chest cavity. And that's going to have the same effect as, say, this syringe here that's being drawn downward, filling this space. All right. Now, the part of the system in your body that is tantamount to that little opening there through which air will pass, of course, would be the nares up here in your nasal cavity. Or if you're breathing with your mouth, you can take it in that way as well. All right, so we've reduced the pressure, the diaphragm contracts, flattens out. Also the muscles here in between ribs, these intercostal muscles, some of those, not all of them, but some ones of them are positioned in such a way that when they contract, they cause the ribs, which normally are kind of directed downward like this, to be pulled up, to be moved up. And when that happens, that actually also adds volume to the chest cavity. You expand your rib cage, thereby expanding the volume of the cavity within. So this relationship between pressure and gas volume, lower pressures cause an increase in gas volume, higher pressures cause it to be reduced. That's called Boyle's law. So with this mechanism that we've got involving our rib cage and the intercostal muscles and the diaphragm here below, that mechanism is exploiting Boyle's law. All right. By the way, the amount of pressure, remember I said pressures are often measured in units of millimeters of Hg or mercury. The pressure that is bearing down on your body when you're at rest in between breaths is 760. Well, it's bearing down all the time. It's the atmospheric pressure. And at sea level, which, you know, Memphis is pretty much sea level, New Orleans, we're just a little bit above that. The pressure bearing down on you is enough to push 760 millimeters or almost a whole meter of mercury up a glass tube. All right. So it's a fair amount of force, although you're not aware of it because you're conditioned to dealing with it. And so if you can create a pressure inside of this space by flattening out your diaphragm and raising the bones in your rib cage up, if you can get a pressure in here that's less than 760 millimeters, air will passively rush into your upper respiratory tract and then into your lungs. Okay, so there's inspiration. Here's the diaphragm flattening out. Now it's down here. Here's the rib cage being drawn outward, and the lungs then fill. That's because the pressure inside of them was lowered. It's dropped down to 758 millimeters of mercury versus 760 on the outside. So a vacuum was created, a negative pressure was created. If you take 758 and subtract 760 from it, you get -2, all right? That's the pressure differential between these two states. And in that condition, air is going to rush in. By the way, the trigger for diaphragm contraction are some nerve endings that project to the diaphragm in much the same fashion as the vagus or accelerator nerves project to the heart. We have phrenic nerves that come off, and they will deliver motor messages that trigger motion in the diaphragm. And they do that both subconscious or unconsciously, you're not aware of it, or if you take over and want to modulate that breathing rate on purpose for some reason, if you want to breathe faster or slower and be conscious of that, you can send signals that likewise would pass through the phrenic nerves. Your brain would generate those thoughts, the need to breathe faster or slower. That information is going to project to the diaphragm by the same route. All right, so yeah, this shows the involvement of the intercostal muscles. And also, I didn't mention it, I forgot, but beneath your main pec muscles, your pectoralis major muscles, you got some smaller pectoralis minors, and you can see they insert on a couple of ribs up here in the rib cage. So when they contract, they lift the rib cage up. These intercostal muscles positioned in here, you see how their fibers are arranged? These external intercostal muscles, when they contract, they're going to help to lift the ribs as well. That's going to cause, some of the inspiration that's going on. Oh, and then we also have the sternocleidomastoids. These big cylinders of muscles in your neck right here. Those are sternocleidomastoids. And in inspiration, especially when it's forced, like if you're having to breathe really hard because you've done a lot of work, those will get involved there as well. Usually at rest, it's just your diaphragm and maybe the external intercostals to help out a little bit. But in really forced situations, say after a big sprint or something, the pectoralis minor and the sternocleidomastoids will get involved as well. All right, so those are the major events of inspiration. The phrenic nerves bring the impulses to the diaphragm. They start to contract and flattens out and increases the volume in the chest cavity. At the same time, you got your external intercostals and perhaps pectoralis minor and sternocleidomastoid helping as well if you really need extra help. And that's going to help to generate the negative pressure in the chest cavity so the air just passively rushes in. Expiration kind of reverses that. And so the pressures will go back up and be about the same as the surrounding pressure. So there's no impetus for the air to be brought in. And so one of the things happens is when you expand a lung, in the tissue that makes up the outer cover of the lung, just beneath the visceral pleura, and in other places within the lung, you've got some elastic kinds of tissue there, elastic connective tissue, that when it gets stretched, will spring, back inward, to oppose that stretch that happened. So it's like you draw a rubber band out or a balloon or something, it's stretchy, you pull it out. When you let go of that, it recoils, right? And so you can expect some elastic recoil to happen. Just blow up a balloon sometime and then don't tie it, but just let the air run back out. That's elastic recoil, right? Sometimes you can take that opening of the balloon and kind of draw it tight and have air pass out through it. That is an analogy for how vocal cords work, right? You can make this little squeaky sound. That's like passing air over vocal cords in your own upper respiratory tract. So that's elastic recoil. The other thing that's helping is what's called surface tension. The alveoli have this sort of tension in them that derives from the fact that there's a lot of water in a surfactant sort of fluid that covers the inside of each one of your alveoli, and it creates this surface tension. So there is a little bit of a tendency for the alveoli when they're stretched tight to recoil as well. And we'll talk about that surface tension and how sometimes it could be problematic. There are times when it can lead to very serious consequences if there's something that is not present to sort of oppose it. Can lead to lung collapse sometimes. But in most folks, the small amount of surface tension that arises in the alveoli is not enough to cause that, but does help with expelling air during expiration. All right, so yeah, at the height of expiration, the pressure inside of your thorax, where your lungs are, is actually going to be above 760. And that's going to cause the air to be pushed back out, right? Whereas if the pressure was exactly 760 within your chest cavity, it would exactly match the pressure outside. There's no air entering or leaving. You've kind of got an equilibrium going on there. Now, there's a way to force expiration, just as there is a way to force inspiration. The muscles involved are fewer, but there are what we call internal intercostals, and there are abdominal muscles that come into play. Things like the rectus abdominis and so forth can help. Oh, and here's another aspect of recoil that I forgot to mention. So the diaphragm is positioned above the liver. It's bulging upward here. This is when the diaphragm is relaxed, shaped like a dome, and the liver and other organs beneath it are going to kind of push up against that. And so when the diaphragm contracts, it pushes these organs down. And they're fairly firmly attached by mesenteries and things to the body wall, so that when you release that force that's being placed on them and relax, the diaphragm springs back up into the thorax, and the organs do as well. They push upward, and they're going to help you to breathe out, right? All else being equal, I think it's a little bit easier for you to expire efficiently than it is to inspire or breathe in efficiently. But we talk about recoil, right? That's an important aspect of it. And here are the internal intercostal muscles, and the way their fibers are oriented. When they contract, they're going to pull down on the ribs above them and lower the volume in the chest cavity, right? Let's look at the external intercostals, see how they're oriented back this way. When they contract, they're going to cause the ribs to lift up, as these arrows in here indicate. All right. When the internal intercostals contract, they're going to cause expiration. Just remember, those are opposite one another. Internal contraction leads to expiration. External contraction leads to inspiration. All right. Hopefully that makes sense. All right, and so there's the major events in expiration. All right, so this is something here, when we get beyond that point, we're into discussion of what are called volumes and capacities. I think what I'm going to do is pick that up next. Actually, when we meet in lab Wednesday, we're going to go over this. And so I'm going to go ahead and stop the lecture right here at slide number 33. And so when we meet again on Wednesday, I'll be able to jump in right here and talk about all of these things. Yeah. So we got about 33 more slides to go after the one that I'm ending with today. All right. So I'll see you all on Wednesday. And like I say, take notes on this first part, pay attention to it, watch it all the way through, because I'm not going to lecture on that again. All right.